Welcome back to this course on organic chemistry in biology and drug development. We have finished the part 1, that is the biochemistry part. Organic chemistry in biology means basically biochemistry, and in the drug development area we have also studied a number of topics, and the last topic that I had taken up was the polyketides, because we are studying the antimicrobial agents, and under the heading of antimicrobial agents we have different antibiotics, and then we have done how these antibiotics are made, specifically the biosynthesis of penicillin. We also have done how the bacteria acquire resistance against these antibiotics or antibacterial agents, and then we went on to a very important class of compounds which are called polyketides, and polyketides molecules are the characteristic feature of polyketides or secondary metabolites which follow the polyketide biosynthetic pathway. They are having either alternating COCH2 group, or they could be having a double bond like CH double bond CH, or they could be having all saturated carbon framework, but all these 2 carbon units are derived from a ketomethylene. So, basically it is ketomethylene which alternate, but the status of oxidation of the carbon bearing the carbonyl can vary. It can be carbonyl, it can be hydroxy, it can be double bond, or it can be even saturated one. Okay. Now, I told you about that there are different types of polyketides, polyketide synthesis. One is pKs 1 type 1 and then pKs type 2, that is the major classification. There are pKs type 3 also possible. pKs type 1 is basically virtually a single enzyme containing different modules, and each module has got different domains of activity. Okay. So, first be very clear that it is a single enzyme with different modules. Basically, it is like the different the compartments of a train. So, each compartment is basically a module, and each seat inside the compartment is basically the domain, okay, where you sit. So, in polyketide synthase 1, you have this modular system, one particular enzyme, a big protein, it should not call it as it is a multifunctional enzyme, and then you have different modules, each module has different domains. And then there is type 2, what you have? You have different, the proteins are all individual, that means these enzymes are not attached with each other to start with, but at the time of synthesis they come together and then react in a cooperative manner to synthesize the polyketide. Okay. But we are basically discussing the pKs type 1, which is the modular type of synthesis. Okay. What happens? I told you about that there should be a loading module to start with, a loading module. Loading module is the module that is where the units, the building blocks gets attached first, and then that is transferred to the first module. Okay. In the loading module, what you have? You have an acyl transferase and you have an acyl carrier protein, that is your loading module. Okay. And then the so acyl transferase puts the starter unit, because there are now 2 units that react with each other. One is a starter unit which only participates only once, that is in the beginning, and there is what is called the extension unit. Okay. That extension unit repeats itself over and over again depending on the number of carbons that the microorganism or whatever system we are talking about that wants to put. Okay. So, depending on that you will have this the number of extension units. Okay. So, you have a, so start with what it says here, each type 1 polyketide. See, this will be little bit repetition of the last session's talk, because it's little bit, it's easy but little bit complicated, and it's entirely new. Usually it is entirely new to the student. So, I am spending more time on this. Type 1 polyketide synthase module consists of several domains. I have already explained what are domains, and then the important thing is starting or loading module, that is an acyl transferase and an ACP. Then you have elongation or extending modules, where the elongation unit or the extension unit, sometimes it is called elongation unit, that means you extend the chain, that is why it is called extension unit, or you elongate the chain, so it is called elongation unit. Okay. So, that elongation or extending modules comprise ketosynthase, that is obligatory, that has to be there, an acyl transferase and an ACP. These three have to be present in the module. Apart from that, within third bracket it is written ketoreductase is there, enoyl reductase, dehydratase. Again I repeat, you should not worry about the sequence how it is written. The important thing is that what type of functionality or domain that is present. So, if these may be present entirely, they may be absent, or one or two may be present. Okay. So, ketosynthase, acyl transferase, ACP are the obligatory domains in a particular module. Apart from that, you can have ketoreductase, you can have enoyl reductase, dehydratase, you can have enoyl reductase. Okay. And then this is repeated over and over again. Okay. Module 1, then it goes to module 2, then module 3, module 4. Again I repeat, depending on the number of extension units that the microorganism wants to put, the living system wants to incorporate. And then when the desired length of the polyketide has been made, then what happens? There is a termination domain, because you have to terminate the process, and that is called a thioesterase, because at the end of it the product is a sulfur in the form of a thioester, which is linked to the ACP, which is linked to an ACP, and then you have to release it from the ACP, acyl carrier protein, either by hydrolysis. So, it ends up as a carboxylic acid, or it could be a cyclization, that one of the internal, there could be intramolecular acylation, trans-acylation, which can result in the formation of cyclic system. Okay. So, that is basically there are these three are there important things. Then what are the domain activities? Acyl transferase, I already told you, acyl carrier protein that has got an SH to hold up the your the extension or the starter unit. Then ketosynthase, that is the major reaction which carries out the Claisen type condensation via a decarboxylation process. Then you have a ketoreductase, dehydratase, enoyl reductase. These are not obligatory, depends on the nature of the product that the living organism wants to make, and then there is a thioesterase. So, this repeats, and then finally the thioesterase comes. So, that is, so it is not difficult at all. You can actually do retro biosynthesis. If I give a natural product, you can do a retro biosynthesis. Just some characteristic features. This is basically repetition. The starter group usually acetyl coenzyme A, but again that may vary also. It could be propionyl coenzyme A, as you have seen in case of erythromycin biosynthesis. Okay. The starter unit was propionyl CoA and the extension unit was methyl malonyl CoA. Okay. And so, that is the AT and the ACP domain. Then elongation stages, we have already discussed all these things, elongation, and finally this cycle is repeated for each elongation module, and the termination stage when the desired length is obtained. Okay. So, now let us do some let us exercise, okay, that retro biosynthesis. You know what is retrosynthesis. Now, this is what is called retro biosynthesis. Now, these are the two polyketides that are, they are natural products and they are biologically quite active. basically they are some of the toxic agents that are present in many of the cereals or the staple food that we take, like wheat, maize or rice, all these things. So, they have toxic effects. So, and so these are important because your cereal should not have any of this toxic agent. There is one more very toxic agent, that is what is called aflatoxin. Okay. But anyway, we are not talking, aflatoxin is a bigger molecule, that is also a polyketide, but we should take jump step by step. Okay. Now, if I want to first take this one, okay, this is a polyketide. How do I know? Is there any signature that after writing the natural product you see that whether it is a polyketide? Usually polyketides have lot of oxygens in it. There is no hard and fast rule, but it's basically an intuition that there are a lot of oxygens that will be present in the molecule, and these oxygens usually have 1-3 relationship. They are having 1-3 relationship. Okay. So, I see there are a lot of oxygens, there is oxygen here, there is oxygen there, there is oxygen here, there is oxygen here, but there is lack of oxygen in many of these carbon framework, that is true. Now, the polyketide synthesis, if you again analyze carefully, the terminal the thioester which is hooked up with the ACP, that ends up either as carboxylic acid or that ends up in the form of a lactone. Okay. So, you try to find out where is this carboxylic acid, where is the carboxylate function. So, you see the carboxylate function is here because this is an ester or a lactone. So, you can break at this point. So, if you break that, that is the starting point. So, if you break that, what you have is COOH, a carboxylic acid, and then you have this double bond, then double bond O, and this is the methyl, and you have OH. Okay. Now, you work, so there are 2 OHs here, OH and OH. Now, you work backwards. Okay. What will happen? See, this is your basically CO SACP, the final before the hydrolysis, and then you start from there and then work backwards. So, you have a CH2, you have a CO, just put whatever is the redox level, don't think too much about that. If you see OH, put a carbonyl here for the time being, and then there is another oxygen here, put as a carbonyl, and then there should be a carbonyl here by rule that 1-3 position there would be oxygen. So, there must be a carbonyl, it was there, but somehow ultimately through some condensation reaction that carbonyl is no longer there, and then you continue. So, there should be a carbonyl here, then there should be 1, 2, 3, yeah, there should be 1, 2, 3. So, then a carbonyl already present. So, there must be a carbonyl here 1-3 position to match that, and then again after the methylene you should have a carbonyl here, and then you have what, methyl and OH. So, OH means carbonyl. Okay. Maybe the SACP is making the problem. So, there is a methyl here, this is a SACP, and this is the methyl that is present here. Okay. So, now this is so the starting unit is what? Starter unit is, or the starting unit is this acetyl coenzyme A. Then you have how many extension unit? This is 1. Maybe take another color, I think that will be better. So, this is one extension unit, this is the second one, this is the third one, this is the fourth one, this is the fifth one, this is the sixth one, and this is the seventh. Okay. So, that is how you let us see whether this is CH2CO, okay. Then this is CH2CO, then CH2CO, then actually this should take this one, sorry, there is some, I just erase this part, yeah, this is, so that is why, let me see. So, what will happen, this is CH2CO, then you have CH2CO, then you have CH2CO, then you have CH2CO, and then you have CH2CO. Okay. So, how many extension units are there? 1, 2, 3, 4, 5, 6, 7 and 8. So, that means there are 8 condensation reactions that will happen, and if it is a type 1, if it is synthesized by a type 1 polyketide synthase. So, you can now write the modules. So, the first module is, I will just write few of them, AT and then ACP, that is the loading module. Then the second module, remember it all starts from here, the reaction starts from here. So, the second module means you have gone up to this point. Now, what is in the actual, you have seen SCH2, that means the not the second, this is the first module, sorry, the first module, this is the loading module. So, first module I said that each module should have AT, should have a keto synthase right after the ACP, then you should have AT, you should have ACP, and whether you should have other ones or not. Because this ends up as a CH2, that means it should have keto reductase. So, this pen is little bit thick, that is the problem. So, you should have a keto reductase right here, you should have a enoyl reductase, and you should have also a dehydratase and then enoyl reductase. Again the sequence does not mean much, it is just the activities, that is what is important. Okay. So, this should be the first module. The first module should have all the possible domains that are required, because it ends up as a saturated system. So, it should be reduced the carbonyl. So, initially it will be carbonyl, but then it will be reduced then the alcohol is dehydrated, and then the enoyl compound is reduced by enoyl reductase. Okay. And then, but all along it is held up by the ACP. It is ACP is holding the whole chain with a carbonyl at the beta position, and then first it shows it to the keto synthase, and then it is reduced, then it takes to the dehydratase. So, it is dehydrated, then it takes the molecule and shown it to the enoyl reductase, and then the enoyl reductase reduces the double bond, then it then the job of the first module is over. So, now the ACP will transfer that whole chain into the second module, and each module, if you look at the modules of erythromycin, I can show you each module, I do not know whether it is visible, but let us see, each module starts with a keto synthase and ends up with the ACP. So, what happens, and in between there are these acyl transferase, then this keto reductase, all these things are present. Okay. So, what happens, first the loading module transfers it to the keto synthase, reaction takes place between the ACP and the keto synthase, the whole chain is held up here, and this is wedged because there is a keto reductase here. Then it is transferred to the next keto synthase. So, all modules end up with ACP and starts with a keto synthase, they are side by side, so that the ACP can transfer it immediately to the keto synthase of the next module. So, I will not proceed any further, it will just I will not complete that. By this way you can tell what is the second module. In the second module, check what is the status of the carbonyl, and you see the carbon is having a carbonyl. That means, second module has what? Has a keto synthase, then acyl transfer, that is there, and last there should be an ACP. Is there any other domain? No, there is no other domain, because it is a carbonyl. And then the next domain next module, not domain, the next module should have again all three, because it is CH2. And after that, the module, that is the, this is the first module, this is the second module, that is the third module, the fourth module now. There is this double bond. So, what will happen now? That means, it is now present as a, so ketoreductase is present and dehydratase is present. So, by that way you can complete the entire biosynthetic gene sequences, and along with the entire the gene, not the sequence, entire biosynthetic gene, and then also assign the protein that is produced from them, okay, that is actually translated from them. So, this is the way you do it. If you want to do for this compound, this is another metabolite, secondary metabolite, again you find out where is the carboxy, again there is a lactone here, that means there is a carboxy here. So, that will be what? That will be a carbonyl, this will be another carbonyl, and then it goes like this. So, then the next carbon is this one. So, that is there, this is the other one. So, by this way then there is OH, then there is this one CH2CO. So, like this is the SCOACP, and then you have CH2CO, then CH2CO, then CH2CO, 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 CH2CO. Okay. Only you have to find out what is the status of this, what we are assigning carbonyl, ultimately in the metabolite what is their status. I could see that if you break these bonds, actually they are all carbonyl, because phenol, this is enolic group. Enol means actually that is the other form of the carbonyl, right, other form of the carbonyl. So, when you see an aromatic ring with a wedge, that means actually that is a ketone functionality, that is not a reduced carbonyl functionality, because phenols means actually enols, and enols means it is the tautomeric form of the ketone. Okay. So, this way you can work out the biosynthesis of a natural product. If some natural product is given to you, the first thing you see what are the oxygenation pattern, and then if there are a lot of oxygens, so you might think of the polyketide, and then if it is a polyketide, then try to put the oxygens at the proper position, identify the starter unit and the extension unit, and then you can finally work backwards and find what are the starter and the extension unit. How to prove that that is the extension unit, that is the starter unit? What they do? They put C13 level starter and then find out where it ends up, or you find C13 level extension unit and then do by NMR study of the secondary metabolite. You can say that whether that extension unit has been incorporated or not. Okay. So, that is just by simple NMR technique, but you have to use labeled, isotopically labeled your starter units or extension units. Okay. So, that is all about the polyketide synthesis. I think that is the end of the antimicrobial agents. Now, we will go to another topic, that is not microbes, because microbes are organisms which can amplify by themselves, which does cell division and then which can grow by themselves, provided sufficient food is added from outside, or it can extract the food from outside. Another very important small tiny entity which also infects, cause infection in the body. Microbes do that. Microbes include bacteria, include fungus, fungi, but then parasites of course are included in that. But apart from that, you have something which is called a virus. You all know this virus, and we get viral infection quite often. And the question is, virus actually do not fall into the group of microbes, because microbes by themselves they are capable of multiplying. whereas virus cannot multiply by themselves, they need a living organism to replicate and then ultimately do multiplication or amplify new virus particles. Okay. So, a virus is nothing but a small infectious agent that replicates only inside a living cell of an organism. So, there is a difference between microbes and this virus. Okay. Virus are non-living as such outside when they are left alone, but if they enter inside the body, they utilize the machinery of the host and then try to and then grow. Okay. So, these are virus, and so virus can infect all types of life forms. They can infect animals, even plants are susceptible to viral infection, and plants to microorganisms including bacteria, archaea. Humans, we suffer from more of viral infection than bacterial infection, to be honest to you. Now, the question is, how do you design antiviral agents? Okay. Obviously, viruses life cycle will be entirely different from the microbes, because the microbes I have shown you that there are different targets. You have the cell wall, you have the membrane, you have the nucleic acid, you have the proteins, you have the metabolites, all these are there. You have transcription, you can stop the translation process, all these things are there. But in virus, those type of mechanism are basically hijacked. Those mechanisms by which things multiply, they mostly belong to the host. So, if you try to stop those processes, basically you are interfering with the host's own mechanism to multiply. So, that is what is the problem, and that caused much delay in developing antiviral compounds. So, our next basically next session we will talk about how to develop antiviral agents, but now before we end this session, we would like to know what are the different types of virus, how they what is their life cycle, how they multiply when they get inside an organism. Okay. I think these are viruses are different types, but the one common thing is that all viruses have what is called an envelope, an outside coat made up of glycoproteins, and then there is something, a circular thing which is called, this is called capsid, and inside the capsid, so this is called capsid, and inside the capsid you have the nuclear material, means I am not saying it is DNA, you have the nuclear material, okay, which helps it to multiply by hijacking the host organism. Okay. Now, these are this is GP, glycoprotein. These are very important because the glycoprotein actually recognizes if a virus enters into my body, the virus particles are first recognized by my cell, the cells that are present inside my body, and these glycoproteins help this recognition process. Okay. Our cells are all having receptors, we are exposed, we know now what are receptors. These are actually basically present in the membrane of the cell, and with the receptor moiety pointing outwards to the extracellular side. Okay. So, there are different types of interactions that can go on to this receptor side. So, the virus particle comes and sits here, but the recognition is through the glycoprotein. So, envelope is very important, and then inside there is again another coating, circular thing, which is called capsid, and the capsid is enclosing the nuclear material and along with some proteins. So, inside there is this nuclear material and plus some, not many, but some proteins are there which are essential for viral replication. Okay. Now, depending on these nuclear material, viruses are classified. Okay. Viruses are classified like, if the nuclear material is DNA, then that is called a DNA virus. If the nuclear material is RNA, then that is called an RNA virus. Okay. And if the but RNA virus, there is a special type which is called retrovirus. I will come to that retrovirus, but let us try to find out what is a DNA virus. So, DNA virus means, the name suggests that there will be DNA will be the nuclear material. Okay. Now, the DNA, whether it is present in double strand or single strand, both are possible. So, you have double strand DNA virus, you have single strand SS, so DS DNA virus, or you can have single strand DNA virus, that is also possible. RNA virus, usually they have a single strand, but RNA is the genetic material. So, I can say nuclear material, I can basically this is the genetic material, means without it the virus will not be able to do anything, material, okay. Genetic material. So, if the genetic material is DNA, that is a DNA virus, if it is RNA, then it is RNA virus, and in retrovirus, this is also the genetic material is RNA, but I will say what is the difference between RNA virus and retrovirus, that will be in my next slide. In the DNA virus, what happens? This is the, see, the entire virus particle is called virion, that is called virion. The virion is having this envelope and with some receptor sites, means that is for the recognition, and then it binds. See, this is the receptor on the cell, this is the entire cell, host cell. So, this is recognized by this, and then by a process what is called usually what is called endocytosis, it enters the virus, not the whole virus enters, it is only the capsid portion that enters, okay, enters into the cell. And now, if it is a DNA virus, then that capsid goes off, the DNA is injected into the nucleus of the host cell, and this DNA usually there are exceptions, usually that now that can become integrated with the DNA of the host. That means, the host has DNA like this, and if the virus has a DNA which is blue color, suppose, then the virus will, what the virus will do? It integrates the viral DNA into the DNA strand. That is one way. The other option is that it does not integrate, but it utilizes the host's DNA polymerase to make the replication, to have the replication of the DNA, and then it can then form the mRNA by utilizing the host's RNA polymerase. So, basically the virus first is recognized by the cell, the envelope falls off, it goes inside with the capsid falls off, the DNA is injected inside the nucleus, and then there are two possibilities, either that DNA now does not integrate with the host DNA, or it can integrate. These are the two possibilities. If it integrates, okay, from that the normal process like replication and transcription, that will take place. So, the transcription means you are making the mRNA, and mRNA comes out. So, that means this mRNA what is being made will contain some mRNA corresponding to the virus DNA, and they will make the required proteins, and then basically form new virus capsids, and finally, this comes out and an envelope is also formed around the virus. So, DNA virus has basically it is a direct way that DNA goes in, it can integrate or it cannot, it may not integrate, remain in the nucleus, and then by the nuclear machinery, that means the DNA polymerase and the RNA polymerase, the mRNA of the virus is synthesized, that comes out, it is encircled by the capsid, forms all the capsid, means the the required proteins that is made from the mRNA, and then it comes out of the, that is called budding. This process is what is called the budding. Okay. Viral, this is the DNA virus. You have the RNA virus. RNA virus, you have again this, this is the envelope, this is the capsid, and the nuclear, the genetic material is now RNA. So, it goes inside, again recognized and then goes inside. So, that is the capsid along with the RNA, and then what happens, this falls off, the capsid falls off. So, this is now the viral genome, but this is RNA. Now, usually you know that central dogma of biology is that always information flows from DNA to RNA to protein. After the discovery of the RNA virus and the retrovirus, that notion, or what is called the central dogma, that is becoming ineffective here. It does not hold true in case of retrovirus and RNA virus, because in RNA virus the genetic material is RNA, and then RNA, now RNA cannot be integrated into the DNA, that is not possible, because the RNA has to be taken to the DNA, and then DNA follows the typical replication, transcription and translation pathway. So, the virus has a RNA here. Okay. So, first it is the RNA is as acting as a template, the DNA, sorry, this is the RNA. So, another RNA, mRNA is made from, directly made from here. See, you do not have to go to the DNA now, because if you have the RNA, and if it is, remember that when the DNA strands, double stranded DNA is copied, one of the strand remains silent, okay, but that is the coding strand. That means, what is the ultimate sequence of the mRNA, that will be decided by the, that will be the same as the coding strand, okay. But the template strand, that is also called the antisense strand. The template strand has the complementary bases here. Okay. So, now the question is, when the virus has the RNA, what is the status of this RNA, whether it is the RNA of the complementary of the coding strand or the non coding strand, that is important. So, you have, that is why you have two types of RNA viruses, positive strand RNA virus, negative strand RNA virus. Negative means antisense, means non coding. If you remember, one strand which does not act as a template, that is called coding strand, that is also called sense strand, that is also called positive strand. Negative strand is the non coding strand, that is also called the template strand, and that is also called the negative strand. Okay. So, depending on this, what type of RNA is present, positive strand of RNA or negative strand of RNA, that will decide what type of life cycle the virus will be having. I think we will discuss this classification in the next session. For now on, what we know, that viruses are classified into DNA, RNA, virus and retrovirus. Okay. So, we will come back in the next class. Thank you.